Before moving to the Mendel's dihybrid cross, let's first see what a test cross is. Now, whenever a progeny or the offspring shows the dominant phenotype, for example tallness, in order to know the genotypic constitution of the progeny, whether it is homozygous or heterozygous, a test cross is performed. In a test cross, the individual plant of a known genotype is crossed with a homozygous recessive individual, here a dwarf plant. In the first case, where the tall plant is homozygous, when it is crossed with a homozygous recessive individual, that is a dwarf plant, each plant produces only a single kind of gamete, and the next generation progeny, all the plants are tall. Whereas in the second case, where the tall plant is heterozygous, when it is crossed with a homozygous recessive dwarf plant, the heterozygous tall will produce two kinds of gametes, whereas the dwarf plant only a single kind of gamete. The following combinations are achieved in the next generation or the progeny, where half the plants are tall and half are dwarf plant. That is, tall is to dwarf would be ratio 1:1. Now, a cross involving two pairs of contrasting traits is known as a dihybrid cross or a two-factor cross. Let's take an example of a dihybrid cross where we take two characters. First, seeds either yellow or green, and second character, seeds either round and wrinkled. As we can see, in the P1 cross, we take two possible combination of parents. In the first combination, a yellow and round plant is crossed with a green and wrinkled plant, whereas in the second case, a yellow and wrinkled plant is crossed with green and round plant. After the P1 cross, in the F1 generation, all the progeny achieved is yellow and round, and this trait or character is the dominant phenotype. When this F1 generation is selfed, that is F1 is crossed with F1, the following results are achieved in the F2 generation. The F2 generation progeny consists of 9 out of 16 plants that are yellow and round, 3 out of 16 plants that are green and round, 3 out of 16 plants that are yellow and wrinkled, and 1 out of 16 plant which is green and wrinkled. An important point to be noted here is that change in P1 phenotypes, that is the combination of characters of P1 parents, does not affect the result of both F1 and F2 generation. Now, to understand the results of a dihybrid cross, let's consider it to be consisting of two separate monohybrid crosses, where the two traits are inherited independent of each other. And so, the first monohybrid cross will have 3/4 yellow and 1/4 green plants. Remember the 3:1 monohybrid ratio. And similarly, the second monohybrid cross will have 3/4 round and 1/4 wrinkled seed plants. All the possible phenotypes of the F2 generation of a dihybrid cross can be known by applying the product law of probabilities. Here, for first trait of all the progeny of F2 generation, 3/4 are yellow and 1/4 are green. And similarly, for trait 2 of all the progeny of F2 generation, 3/4 plant are round and 1/4 have wrinkled seeds. By multiplying the frequency of occurrence of individual traits, we can compute the combined probability of all the possible phenotypes. As you can see, 3/4 yellow multiplied by 3/4 round, and thus 9 out of 16 plants would be yellow and round. Similarly, 3/4 yellow into 1/4 wrinkled, that is 3 out of 16 plants would be yellow and wrinkled. And similarly, 1/4 green multiplied by 3/4 round, thus 3 out of 16 plants would be green and round. 1/4 green into 1/4 wrinkled, that is 1 out of 16 plants would be green and wrinkled. Finally, the fourth postulate, which is independent assortment. According to this law, during gamete formation, segregating pairs of unit factors assort independent of each other. In further analysis of the dihybrid cross, under P1 cross, a yellow round plant is crossed with a green wrinkled plant, or a yellow wrinkled plant is crossed with a green round plant. In both the cases, different types of gametes are formed, and after fertilization, the result is F1 generation, where all the progeny is yellow and round. Now, in F1 cross, the F1 generation is selfed or self-fertilized. Here, a heterozygous yellow round plant is crossed with another heterozygous yellow round plant. The following types of gametes are formed by each of the parent. Using a Punnett square, we are able to analyze various possible genotypes that are achieved in the F2 generation. By observing the Punnett square, we are able to know that 1 plus 2 plus 2 plus 4, that is 9 out of 16 plants are yellow and round. 1 plus 2, that is 3 out of 16 plants are yellow and wrinkled. 1 plus 2, that is 3 out of 16 plants are green and round. And lastly, 1 plant is green and wrinkled. And hence we are able to conclude the Mendel's dihybrid ratio, that is 9:3:3:1. Lastly, let's see a test cross for two characters. Here, taking the dominant character to be yellow and round, the genotype of individuals showing dominant phenotype have to be found. In order to do that, the plants with unknown genotype will be crossed with a homozygous recessive individual. In the first case, where the plant is homozygous dominant for color of seed and heterozygous for shape of seed, and it is crossed with a homozygous recessive individual, 1:1 phenotypic ratio is found, where half of the plants are yellow and round and half are yellow and wrinkled. In the second case, where the plant is heterozygous for both color of seed and shape of seed, when it is crossed with a homozygous recessive individual through Punnett square, it can be computed that 1/4 plants are yellow and round, 1/4 are yellow and wrinkled, 1/4 are green and round, and 1/4 are green and wrinkled. In the third case, where the plant is heterozygous for color of seed and homozygous dominant for shape of seed, when it is crossed with a homozygous recessive individual, 1:1 phenotypic ratio is also found here, where half of the plant are yellow and round and half are green and round. Fourth and last, when the plant is homozygous dominant for both the characters and it is crossed with a homozygous recessive individual, all the progeny will show the dominant phenotype, that is yellow and round. We shall discuss the Mendel's trihybrid cross together with the Falkline method in a later video.